All right, hello and welcome to this rendition of Global IMMUNOTALKs. My name is Burkhard Becher and I'm hosting today's speaker, and it's a great pleasure to host Bart Lambrecht today. But before I get to introduce Bart, I need to remind you of a couple of housekeeping rules, common things that we do with Global IMMUNOTALK here. As those of you who have seen this in the past will know, there is always a somewhat abrupt ending after Bart's seminar. We will basically stop the webinar and you will be left with all your open questions, but no worries, you can ask your questions on Twitter, and the details were shown to you before in the intro slides, but you'll see them again at the very end of the seminar. You'll see again how to get in touch and ask Bart some questions. And while I'm at it, I also like to remind you that next week, Jim DeSanto from Paris is going to give Global IMMUNOTALK. But enough of that. The most important part and the most exciting part is that I get to introduce Bart Lambrecht, who is here with us today. So thanks so much for your willingness to speak with us today. And allow me to say a few words about what I know about Bart and what, so you all, most of you probably will be familiar with his work, but I still think I should tell you that he obtained his MD in 1993 and his PhD in 1999. In medicine at the University of Ghent. He then became a specialist in pulmonary medicine in 2002, where he did his residency at the Erasmus University Medical Center in Rotterdam, that's in the Netherlands. And he is now a professor of pulmonary medicine, both at the Erasmus Medical Center, but also at the University of Ghent in Belgium. What most of you probably will, or those who know Bart a little bit, will be familiar with the fact that 2012 he became the director of the VIB Inflammation Research Center in Ghent, which hosts more than 400 scientists. And I had the pleasure to visit them once as a scientific advisor, and I have rarely been more impressed when it comes to the state of the art of the environment and the way everything is set up. It's a tremendous achievement, Bart, and I'm, like together with many other people, utterly jealous. So Bart has co-authored more than 400 papers in the field of asthma and allergy and respiratory viral infection. He has been bestowed with so many awards and recognition and prizes that are simply too numerous to name here. And you can look it up, but I think another thing that wasn't necessarily an easy thing to achieve is what he received 2 ERC. This is the European Research Council funding scheme, which is very, very hard to get, but that 2 in a row, it's a very rare feat. I'm sure there will be another one coming up, but he got already 2, so that again is a rarity already and a sign of his scientific excellence. So together with Professor Hamida Hamad, he leads a research unit of 36 people. And the research unit is centered around the role of myeloid cells, phagocytes, in asthma and respiratory viral infection. And they study how dendritic cells and macrophages get activated and how they bridge innate and adaptive immune responses in allergies and respiratory disease, but also respiratory viruses. What I think is possibly one of the most important things to say about Bart is that he and Hamida try very hard not just to understand how asthma works, but also to see whether it can be translated in collaboration with biotech or big pharma to bring their ideas into the clinic and to test this. And I've always been deeply impressed. Now, before I pass on to Bart's seminar, there's one other housekeeping thing that I'm very excited by, and that is the question that we do with all IMMUNOTALK speakers, and that is we ask them a specific question. And the question for Bart would be, could you share one of the most impactful decisions of your scientific or in your scientific career?
So yes, I can, Burkhard, and thanks for the kind words, by the way. So if I look back at my career, and I think, I'm not at the end of my career, but what was really very important for me at a certain point, I was a medic and then made a decision to do fundamental research. That was the first important decision. But I think the most impactful one has been at a certain point, while I was doing fundamental research for about 15 years. I had something like, is this really it? Will I do mouse work for the rest of my life? Even being a doctor, will I stick to mouse work? And I think for me and for the group, it's been very important to make connections with pharma industry and particularly with biotech. So some of us, basic scientists think that once you are in biotech, it's more of a routine job where you have to, like sometimes do maybe paperwork, sometimes the occasional experiment, but there's a lot of negative decisions being made over your head. Well, I have completely the opposite experience. So what I have learned from biotech is that you can really work very focused, very goal-oriented, and there are some very, very clever people in the biotech industry who are really good scientists. And I think, if we as academics can collaborate more with that branch of science, with biotechnology, where it's more goal-oriented, process-oriented, and really where their job is to translate the basic science findings into a next level, ready to bring it to the clinic, ready to bring it to production. I have learned so much from that, also for my basic research. So my advice to young scientists would be, if you're actually considering a career in biotech or pharma, just carefully choose your job. Don't go for a paperwork job, because there's many of them, but choose a job where you can still do research in biotech, and you will be amazed how much opportunities there are also for good level basic science in that job. And I was really happy to be able to do that from the academy side, if you will.
All right, well, that's super interesting. You're right, that's not the most obvious view on biotech and big pharma in general, but I very much appreciate, and I'm sure your people listening to you will appreciate your endorsement. So without further ado, Bart, the floor is yours. I'm excited about your talk. Please share your screen, and the floor is all yours.
All right, so thank you very much, Burkhard, and also to the other organizers for giving me this opportunity. So I will talk about something very unconventional that probably many of you have never heard about. It's the idea that some proteins in our body, proteins that are normally in a soluble state, actually go into a phase transition and become protein crystals. And I hope by the end of my lecture, I will be able to convince you that protein crystals can actually be initiators and drivers of immunity and chronic inflammation, and could actually be targeted for therapy for a number of diseases. So first of all, a couple of definitions. I mainly work in allergic diseases. So allergy is often misconceived. Now, what is allergy in the true sense of the word? It's a genetic tendency to form IgE antibodies against common environmental antigens, where the majority of people would form an IgG response or an IgA response. And an allergen, somewhat arbitrarily, is called an allergen when it induces this IgE. So it's a bit of a circular reasoning. But the presence of IgE antibodies, which is typical of allergy, can be measured in the bloodstream using a simple blood test, where it's just one of the least abundant antibodies in the blood. But it can also be tested using a skin prick test. And I think many of you will have undergone this at a certain period of your life, because nowadays, 1 in 2 people in the Western world is allergic, meaning it has a positive IgE in the blood or a positive skin test to some allergen. And this allergy can manifest into some allergic diseases like hay fever. I think we all know that. It's the reaction, the sneezing, itchiness when you're exposed to tree or grass pollen. But it can also be much more severe. Think of it on the right-hand side here. This is a kid who ate peanuts, had an IgE response to peanuts, and the child is severely anaphylactic with swelling around the lips and the throat, and this can lead to death in no time. But I want to stress out that this is just a possibility. So having an IgE response in your blood is only a risk factor. It's not necessarily going to lead to disease. And we think that currently maybe 1 in 3 people in the Western world has an allergic disease. And for asthma, for example, it's 1 in 8 people who have asthma, mounting up to 300 million people worldwide actually who have asthma. So it's more than just IgE. If you take biopsies from people with asthma, and this is a biopsy we took years ago, from an asthmatic patient, you will also see that in this form of allergic disease, there's often eosinophilic inflammation. And eosinophils are hallmark cells found in so-called type 2 inflammation. Type 2 meaning that it's controlled by Th2 cytokines. And eosinophils are some of the most toxic cells in our body. So they're really meant to kill parasites. So they have a lot of enzymes and toxic products like peroxidases and neurotoxins that can kill large extracellular parasites. But they also produce lipid mediators and cytokines and other proteins that can cause damage, not only to helminths, but also to tissues. So in a lot of allergic diseases, you have eosinophilia as the hallmark inflammation. And these eosinophils are driven by very often an underlying adaptive Th2 CD4 immune response. And this is a biopsy that we took years ago from a patient with asthma, and it's immunostained for CD4, where you see immediately that in the bronchial biopsy, it's really full of CD4 Th2 cells, T cells here. And this is more in a schematic manner. This is summarizing years of research by numerous labs. And it's now clear that in allergic type asthma, so where you have a person who is allergic, say, to house dust mite, for example, you take an asthma biopsy or you look in a mouse model for house dust mite allergy, there's an important role for dendritic cells capturing the antigens and then presenting these antigens to Th2 cells. And then those Th2 cells will produce a couple of cytokines that are very typical for a Th2 response. So they will make interleukin-5, driving the tissue eosinophilia, but they also make IL-4, driving IgE synthesis, and they make IL-13. And IL-13 induces the production of a goblet cell metaplasia, so leading to overproduction of mucus. It will cause contraction of the small airways or bronchial hyperactivity. It's the tendency of the airways to react to cold air, for example, and it will prime the vessel wall for extravasation of more inflammatory cells. So those Th2 cells are pretty crucial cells. And in a mouse model, for example, if you get rid of Th2 cells, for example, using an IL-4 receptor alpha knockout, you will have no asthma. And if you see this on the right-hand side, this is a piece of autopsy sample that's taken from a person dying of asthma. You actually see that the airways here are narrowed because there is mucus production, there is inflammation, smooth muscle contraction, and there is also some remodeling. You see here, for example, that the smooth muscle layer is way too thick and also the basement membrane is thickened. So these are the hallmark signs of asthma leading to airway narrowing. So how can allergy be a risk factor for asthma? So it's a risk factor because on the one hand side, you will arm your mast cells and your basophils with IgE. And then when you see an allergen, those mast cells and basophils will fire and they will release very toxic substances like histamine, leukotrienes. And this causes itchiness in the eyes and in the nose, but it will also cause plasma extravasation in the lungs, for example, leading to the beginning of inflammation. Second, and most important reason why allergy contributes to asthma is that dendritic cells in asthmatics are also armed with high affinity IgE receptors, and IgE will be used to capture allergens so that these allergens are presented better to Th2 cells, and then those Th2 cells, through their cytokine production, will activate eosinophils and ILC2s, etc., leading to chronic ongoing inflammation. And then the third, less known reason why IgE leads to asthma is that when you have IgE in your blood, your plasmacytoid dendritic cells will also armed with IgE. And when then an allergen cross-links that IgE, it completely cripples the production of type 1 interferon by your PDCs. So those people are very, very sensitive to viral exacerbations because they don't produce adequate amounts of type 1 interferon. They will be sensitive to viruses. And this is the most common scenario. An asthmatic coming in twice a year with an asthma exacerbation, meaning the asthma gets worse. And this is caused by respiratory viruses that, induce extra inflammation on top of the underlying type 2 inflammation that you see in asthma. So for a long time for many doctors, asthma was a simple disease. It's a disease. It was assumed to occur throughout the lungs. And this is the narrative we tell to patients. It's everywhere in your lungs. It's driven by inflammation, and it's driven by smooth muscle contraction around your airways. This leads to a narrowed airway compared with a healthy lung. So we need to treat you with a combination drug containing an inhaled steroid to treat the inflammation and a bronchodilator to relax the smooth muscle, and you will be fine. So this is how 20 years long we used to think about asthma. But then when those drugs became really wide used, we started actually realizing that not all asthma is actually sensitive to steroids. And there's a significant proportion of people where steroids will not reduce the symptoms of asthma nor the airway narrowing. And also the immunology of that form of asthma has been completely unraveled over the past years and has led to a lot of new drugs. So that form of asthma is often in elderly people. It's non-allergic asthma. And rather than being driven by allergen recognition and Th2 cells, it is driven by innate lymphocytes type 2. So these ILC2s look completely like a Th2 cell, but they don't have a T cell receptor. Yet they respond to epithelial cytokines like IL-33, IL-25, and TSLP. And then this will lead to the production of the same cytokines, IL-4, IL-5, IL-13, and also a little bit more interleukin-9. So because these ILC2s, in contrast to dendritic cells, are very steroid resistant, we had to come up with new drugs. And this is where a whole class of new drugs has been developed. We can now block interleukin-5 with a drug. We can block the IL-5 receptor, and we can block IL-4 and IL-13 with an antibody that blocks the IL-4 receptor alpha called Dupixent. So because of this insight into the immunology or basic immunology of asthma, we have now come up with new drugs that are not corticosteroids, but that target the individual effector cytokines of asthma in those people with non-allergic asthma. They would also work in allergic asthmatics, but it's way too expensive to do that. An allergic asthmatic can better be treated with immunotherapy or with an inhaled corticosteroid when things go well. So this brought us over the years to the importance of the epithelium. So as mentioning already, that innate lymphocytes type 2 get activated by epithelial cytokines, but we've also come to realize, based on our research, for example, on house dust mites and on farm dust and on air pollution, that the epithelium is a very, very crucial sensing mechanism for producing cytokines. And then those cytokines not only activate innate immune cells, but they will also activate the dendritic cells, and particularly the type 2 dendritic cells that drive Th2 adaptive immunity, and the other cytokines will drive innate immunity. So in most asthmatics that come to the clinic, there's always a combination of the adaptive type 2 immune response with an innate type 2 response made up of ILC2s, basophils, eosinophils, and alternatively activated macrophages. And again here, these insights have rapidly led to new drugs. I used to work on TSLP, like 25 years ago, when I was still working on Rotterdam. We thought it was going to be nothing. I'll tell you, TSLP is one of the most promising drug targets, and it's now been registered already in the US and in Europe as a new drug for treating asthma. So you can block TSLP. IL-33 blockade is also coming. And this is all based on the insight that the epithelium is the driver of type 2 immunity in asthma. There is, however, another reason why, and that's the remainder of my talk now, there is another reason why not always steroids are the best choice for treating asthma. So rather than thinking that asthma is a generalized disease, we much more have the idea now that asthma is a disease that occurs in discrete hotspots. And these hotspots are found around areas of mucus plugging. So this is a patient here who died of asthma, where you see here, there is this mucus collection sitting in the airways. And this is a staining here that Helena Eigerter did in our lab in collaboration with John Fahey and UCSF. It's an asthma autopsy case where it's stained for mucus. And you see that some of the airways are completely obliterated by airway mucus. The right top hand side here, this is something that came out by bronchoscopy. So it's a complete cast from the inside of the lungs. And this is by no means a new finding. There is reports going back to the 1700s of a ship's captain that came back on a boat in Amsterdam, and they thought that he was coughing up the lung vasculature. And it turns out this was actually a complete cast of the bronchial tree that was coughed up by this patient with asthma. So these plugs, when you have them in your airways, you can imagine that there's not going to be any airflow, and it's going to be dramatic in terms of lung function, irreversible airway obstruction. So we've gotten very, very interested in understanding how do these plugs form and what do they actually do in ongoing asthma. So, and for this, I have to bring you back again to history. And already in 1870, two doctors simultaneously described that in the sputum and in the mucus plugs of patients with asthma, you can find crystals. You see here on this simple drawing here on the light microscope, you see plenty of crystals here on a sputum sample. And also in this plug here, you see these bipyramidal crystals. And these crystals have come to known as Charcot-Leyden crystals, after Jean-Marie Charcot and Ernst von Leyden, who discovered them. Charcot was a neurologist, von Leyden was an oncologist, and they both, by chance observed this in asthma patients. So over the years, we've come to know that these Charcot-Leyden crystals are made up of a protein. So very weird, because normally proteins don't crystallize. So this is an immunostaining here that we did for galectin-10, and galectin-10 is the protein that crystallizes into Charcot-Leyden crystals. And galectin-10 is made in humans predominantly by eosinophils. So this is the nice link. Asthma is eosinophilic inflammation, and then you see these crystals made from an eosinophil-derived protein in the mucus. And this is here a patient here with asthma that we saw a couple of years ago. You see in the mucus here plenty of eosinophils and also these crystals here in the plug of this patient, just like Charcot described this in 1870. So it's a protein, galectin-10, that crystallizes. And this is from Rosanna Mello. It's electron microscopy. And these are eosinophils that are dying, where you immediately see that in the vicinity of a dying eosinophil, you see these almost surreal structures, which are Charcot-Leyden crystals. And galectin-10 is one of the most abundant cytoplasmic proteins of an eosinophil. This is an immunogold staining here, where you see that the small immunogold particles stain the cytoplasm, but not the granules. And in a higher magnification here, you see that these galectin-10 immunostaining is in the cytoplasm. There are also granules that have a crystalline core, but this is actually a different crystal. This is major basic protein. It's also a protein in an eosinophil that crystallizes, but it's not galectin-10. The galectin-10 is in the cytoplasm. So a couple of years ago, we wondered, how could these crystals be forming? So these are human eosinophils that are triggered using PMA ionomycin. It's a classical inducer in neutrophils, for example, of a so-called netosis, the pushing out of DNA from the nucleus or the mitochondria of neutrophils. So when you look here after 15 minutes, it's in the GAL10 immunostaining. The GAL10 is in the cytoplasm. The nucleus is in DAPI staining. 20 minutes later, you see that the DNA is coming out. And then you see that the DNA nets actually start being decorated with galectin-10 immunoreactivity. And another 5 minutes later, all those nets associated galectin-10 retract into crystals. So crystals form, when eosinophils undergo etosis. So the cytoplasm gets pushed out, and then on the crystal, on the nets, that's where you see crystallization of the galactin-10 into sarcolytin crystals. And even in patients with chronic rhinosinusitis, where you have a lot of eosinophilic inflammation, we could correlate the degree of etosis measured by extracellular DNA, as shown in this picture here, to the number of sarcolytin crystals you can find in a high power field. So they're correlated. So, and then we did something very unconventional. We wanted to understand better how this protein crystallizes inside patients. So we did, with Sava Savides, a colleague in the VIB here, we fished out crystals from the nose of a patient with chronic rhinosinusitis. So we found those sarcolytin crystals and then sent those in vivo grown crystals to the synchrotron facility and obtained below 2 angstrom resolution crystal structure of the protein. So galactin-10 forms a dimer, and in the dimer interface, there is a carbohydrate recognition domain. So there are 2 carbohydrate recognition domains formed in the dimer interface. And there's a little sticky end here, which is a tyrosine in position 69. So the carbohydrate recognition domain, I'll come back to it later. It has a tryptophan pocket here, and it can accommodate riboses, for example, and a number of other sugars on the backbone of other proteins. So normally, proteins are hard to crystallize, but here we were very, very lucky. So with Savas, who's a crystallographer and structural biologist, we decided to produce recombinant galactin-10 in Escherichia coli or in yeast production, and we added a his tag to make it soluble. And as soon as we removed the his tag, the protein started crystallizing into beautiful crystals. And this was so efficient that it was super cheap. It's been calculated that normally to make a milligram of recombinant crystallizing protein, it's about 80,000 times more expensive than diamonds. And in fact, that's why people never do work with crystals, injecting them in vivo or something. It's because it's just way too expensive. But this crystal or this protein crystallizes so easy that we can make bucket loads of recombinant sarcolytin crystal. And just for fun, I've put this picture in black and white here to show you the forms of these crystals that form from the recombinant material is exactly similar to the original drawing of Charcot in a leukemia patient in 1850, where they were using a simple light microscope to describe them. Actually, all those forms can be found in the recombinant crystal fraction that is made in E. coli. And when Savas modeled the crystal packing interface, it's immediately clear that there's a long axis symmetry. So these dimers, they hook into one another in a long axis symmetry through this tyrosine 69, forming a base, almost like a base interaction. So the tyrosine ring is stacking with a tyrosine ring of an adjacent galactin-10 dimer. And like this, you form long linear chains. So it's almost like making Lego blocks. So you click those tyrosine 69s onto each other into long linear chains, and then the longest ones are in the middle, the shorter ones are arrayed next to it, and this is how you form a bipyramidal crystal. And the tyrosine 69, which is like the little nap on the Lego block, is always exposed on the tip ends of the crystal, and this is also how the crystals would grow in vivo. So how do we know this tyrosine 69 is so important? So we mutated systematically the key residues in the crystal packing interface. So this is the wild type protein. So it crystallizes, it forms a pellet in your Eppendorf tube. When you mutate this tyrosine 69 to a glutamic acid, you have as much protein in this tube, but there's no crystals, there's no pellet. So other residues are also important, whereas other predicted residues didn't do anything. So we have a clear idea how this protein crystallizes, but we also have great tools now because we can have galactin-10 in a dimeric form, non-crystallizing, or galactin-10 in a crystallizing form. So we then started asking the question, could these galectin-10 crystals or sarcoidin crystals be more than just markers of eosinophilia? This is how it was in the textbooks. This is how I learned about it in medical school. But now we have these cheap crystals. Can we use them in vivo? And mice don't have galectin-10. That's the caveat, and that's why nobody has worked on galectin-10. It's because mice don't encode it in their genome. So what we did is we injected intratracheally galectin-10 crystals or the soluble gal-10 mutein, and immediately observed that the crystals induce acute neutrophilia, also monocytosis, etc. And this is not the case with the soluble galectin-10. And we also saw cytokine production within 6 hours. We saw the production of IL-6 in the lungs and in the lymph nodes, and this was not the case with the soluble galectin-10. So it's not the dimer of galectin-10 as such. It's really the crystalline state of the protein that determines whether this is a pro-inflammatory stimulus for these mouse airways. Many crystals are adjuvants. Think about aluminium hydroxide. We've done research years ago showing that aluminium hydroxide is also triggering immunity because it's a crystal and because it's releasing uric acid crystals. So we wondered, just like alum or uric acid, if you would mix ovalbumin with sarcoidin crystals, could these things act as adjuvants? And we did this by reading out the proliferation of OT2, ova-specific T cells. So if you inject OT2 cells and then ova intratracheally, in the mediastinal lymph nodes, you will see divisions of CFSC dilution or violet trace dilution. When you add crystals, however, there are more divisions. You see this here quantified. But when you add soluble galectin-10, it's the same as giving ova alone. So in other words, these things work as an adjuvant, but only when they're in a crystalline state. And it's more than that. It's not just T cell divisions. When you re-challenge the mice with an intranasal administration of ovalbumin, harmless protein antigen, only the mice that received ova plus crystals are sensitized, are Th2 sensitized, and they get airway eosinophilia and they get airway neutrophilia. And this is not seen when you mix the ovalbumin with soluble galectin-10. So the crystalline state of galectin-10 determines whether it's an adjuvant or not, breaking inhalation tolerance to ovalbumin. So all of this was done using transfer of crystals or gal-10 to the airways of mice. And remember, the mice do not encode galectin-10 in their genome. So over the past 3-4 years, we've worked hard and we made 10 different mouse strains expressing galectin-10, either in eosinophils or in lung epithelial cells or ubiquitously or intracellularly secreted formats. And we now have mice that are humanized for galectin-10. I'll just show you one example of this. We've given them all kinds of funky names with like the galactic mice, the Galileo mice, the galaxy mice, all based on where the protein is expressed. And this slide here, taken by Helena Eigerter, a postdoc in the lab, is actually the galaxy mice. And the galaxy mice, the name comes from galectin under the CC10 promoter, which is a promoter that's typical for lung epithelium. To make the protein secreted, we also added a signal peptide. So we use the BCL10 signal peptide, which just brings the sarcoidin crystal from the cytoplasm to the secretory pathway. And then we can have high-level secretion of galectin-10 in the airways. And this is exactly what happens. So this is an immunostaining here for galectin-10, human galectin-10 in these mice. And you see immediately that the secretory goblet cells here are producing galectin-10, and that there are also hotspots where sarcoidin crystals start accumulating in the lumen of the airways. So we now have a mouse. One of these mouse here forms spontaneously galectin-10 crystals because of this transgene expression. This is another one. It's the galactic mouse where the galectin is expressed under ubiquitous promoter. We've run in through a mild house dust mite asthma model where the wild types have like mild goblet cell metaplasia. You see pus staining here. You see some inflammation here around the bronchi and around the vessels. But if you take a galactic transgenic animal, what's immediately clear is that this mouse has plugging of the small airways, compared with a non-transgenic animal where you only see goblet cell metaplasia. These mice have way more, mucus plugging, and those mucus plugs contain crystals here. This is a crystal here, for example, in the mucus plug. And if you quantify this, there's more mucus plugging. And just like we saw with the recombinant Gal-10 crystals, we now see that in these mice, there is enhanced Th2 immunity. There's more interleukin-5 production, more IL-13 production, and very importantly, there's more bronchial hyperactivity, the tendency of the airways to contract to non-specific stimuli, in this case, methacholine, which is a bronchoconstrictor. So this is no longer transfer of crystals. These are endogenous crystals that reinforce the severity of asthma and that cause mucus plugging. And this association of crystals with mucus is something we always see. So remember, I told you we were fishing out crystals from the nose of chronic rhinosinusitis patients with nasal polyps, a typical eosinophil-rich type 2 disease, where some of this mucus looks gross. The surgeons actually call this peanut butter or hard clay. If you have it, it's really, really nasty. It can be very, very chronic. But this is exactly where we find those crystals. So this is an immunostaining here from that type of mucus, where some regions are really packed with crystals. And if you look from a surgery piece from that same patient, you actually see that the crystals impinge on the epithelium. So the crystals really go all the way into the epithelial top layer, and we think those crystals are tickling the epithelium. They're causing some physical damage. They're irritating because they're needles. They're actually irritating that epithelium. So to start to study that and to model that, we recruited a new MD PhD postdoc who's coming from France. So that's Dr. Engie Ahmed, who worked with Arnaud Bourdin. And they have actually set up a very, very neat model. So they can take blood from patients with lung diseases, asthmatics. They take the PBMC fraction from those patients and then use Sendai virus factors to express the Yamanaka transcription factors. So they're reprogramming the blood cells to become induced pluripotent stem cells. And then those pluripotent stem cells, after about 100 days, can be turned into pure lung epithelial cells. And this is, to me, really science fiction. So the bottom panel here, you see the PBMC fraction that Engie isolated. You go to erythroid progenitors, you form iPSCs colonies. This starts looking like a blastocyst, you know, like a mouse embryo almost. And then when you differentiate, you can generate lung epithelium with cilia, with mucus cells. The whole thing is actually reproduced starting from the blood of healthy controls or asthmatics. And the whole differentiation process, I won't go too much into detail. This is all published now by Engie, but it's really mimicking the embryogenesis of the human lung in a dish. So you go from stem cells to anterior primitive streak to the definitive endoderm, and normally that's where the lung buds off into the anterior foregut endoderm, and then you really start getting lung epithelial cells. You can then submerge those cultures, and when they're confluent, you expose the top layer to air, the bottom to fluid, and you get these so-called air-liquid interface cultures with beautiful goblet cells, with secretory cells, and with ciliated cells. So when Engie took these cultures and we exposed them to interleukin-13 and Charcot-Leyden crystals, we were absolutely thrilled of what we saw. So for the first time ever, we saw that those air-liquid interface cultures started making these so-called gel-forming mucins. And gel-forming mucins, this is here, for example, taken from a patient with cystic fibrosis, where you see these long threads of Jacqueline-positive mucus that can be found in the lumen of the airways of people with cystic fibrosis. The exact same structures are formed when you expose air-liquid interface cultures of humans with IL-13 and Charcot-Leyden crystals. And the cool thing is that these mucin forming or this gel forming mucins, they're always made up of a combination of MUC5B, which is healthy mucus, if you will, and MUC5AC, which is pathogenic mucus, typically seen in asthmatics under the influence of IL-13. And if you do a higher magnification of this MUC5AC rich, MUC5B rich region, this is exactly where the crystals impinge on the epithelial cells. So we think there is some mechanical trigger on the human lung epithelial cells that causes goblet cell metaplasia and actually causes mucus and unhealthy mucus production to be formed. And you see actually when we compare iPSC-derived cultures from patients with asthma versus healthy control epithelial cell lines made from the blood of patients, of healthy controls, we actually see that the combination of IL-13 and Gal-10 crystals is mainly doing this in diseased epithelium, not in healthy epithelium. So this is showing that there might be some unique sensitivity of asthmatic lung epithelial cells to the effect of crystals to be able to produce pathogenic mucus. So we think these areas are really hotspots for type 2 immunity. And this is, again, work that we did in collaboration with John Fahey in UCSF. So this is an autopsy piece here from a patient dying of asthma, where you see 3 areas where the airways are plugged. And there's also airways which are not plugged. So you see immediately here, this airway is obliterated, this airway is still open. But if you then do an immunostaining for eosinophils, you actually see that the eosinophils, typical sign of type 2 immunity, are mainly found around the plugged airways, but not around the non-plugged airways. So this is leading to the concept that these highly localized hotspots are really niches, if you will. They're specialized niches for type 2 immunity. And you see them accumulating as type 2 immunity becomes more severe. So typically in people with irreversible airway obstruction and with concomitant chronic rhinosinusitis and nasal polyps who are also steroid resistant. So this is what I've shown you so far, is that plugged airways are hotspots, and this is because it starts with type 2 immunity, ILC2s, Th2 cells making interleukin-13, driving the production of MUC5AC and goblet cell metaplasia. Then the eosinophils come in. The eosinophils get recruited in response to IL-4 receptor triggering on the capillary endothelial cells. And then the eosinophils, under the influence of IL-5, again coming from the Th2 cells and the ILC2s, eosinophils degranulate and they release peroxidases. And these peroxidases will cross-link the mucus into a kind of a glue-like structure. And then the second hit is that the eosinophils will undergo eitosis. They will release their crystals, and those crystals get entangled in the mucus glue, and then they will start tickling the epithelium, and that epithelium and the underlying stroma sets out changes in the environment. It could be IL-33, it could be TSLP, that causes a chronic survival benefit for local cells to survive. And we believe those areas are also places where dendritic cells talk with Th2 cells, with resident memory cells, with ILCs, etc., with stromal cells. And this leads to chronicity. And around those plugged airways, it becomes like a self-perpetuating loop. So that when asthma, which initially starts with an allergic trigger acting on barrier epithelial cells to activate immune cells, all of a sudden, when the mucus pluggings come in, the mucus plugs will start triggering the immune cells. And you no longer need the initial trigger, and the whole thing becomes self-perpetuating, particularly around those hotspot areas in the lungs. So obviously we want to model this in the mouse, and we've developed a couple of new mice models. This is again Helena Eigerter and colleagues. So we can now use hydrogels containing hyaluronic acid hydrogels, and if we don't put crystals in there, there's not a lot of plugging. If we put crystals in there, basically everything gets clogged within a couple of days, and we get dramatic airway inflammation. And this is such a plug here. So this is the hydrogel here. You can still see some remnants here of crystals. And this actually leads to neutrophilic inflammation. Remember when I told you about the crystals? They're recruiting neutrophils. And the neutrophils actually get activated. And this is very recent research. So we've seen that the neutrophils that get recruited by the crystals actually lead to netosis. So you can measure 24 hours after injecting crystals, but not soluble galectin-10. You see double-stranded DNA in the lavage fluid. And when we take human neutrophils and we expose them to crystals, those neutrophils throw out their DNA nets. So this is a classical netosis. And then the DNA gets entangled with the crystals. And we call this the barbed wire concept. It's like the barbs are the crystals and the wire is the DNA. So we can also see this in vivo. This is Jasmijn Bergelman, a new PhD student in the lab. So this is mice receiving crystals where you can see the fluorescent crystals here in the lungs. And exactly at those regions, we also see citrin-related H3, a sign of netosis. And this is also where the myeloperoxidase coming out of the neutrophils gets released into the extracellular space. And also with electron microscopy, if we take human or murine neutrophils and we expose them to crystals, you see that the small crystals are eaten by the neutrophils, but the larger crystals really get wrapped with a DNA net because it's kind of form of frustrated phagocytosis. The neutrophils will actually throw out their DNA and get wrapped around the crystals. So how could this work? And remember I told you the carbohydrate recognition domain it's a dual carbohydrate recognition domain. And if you actually look at the structure of the crystal, these carbohydrate recognition domains array along the long axis of the outside of the crystals. And the putative ligand could beautifully bind along the crystals to these repetitive arrays of carbohydrate recognition domains. So we believe that DNA binds actively to the carbohydrate recognition domain of the crystals, forming these barbed wires. And why do we believe so? Is because we mutated the pocket of the carbohydrate recognition domain. And this was Kenneth Verstraete doing this in Savas Savides' lab. So we were mutating this tryptophan 72. So if you take the wild type crystals and you stain them with DNA and DAPI, you see the crystals. But if you take crystals with this tryptophan 72 mutation, you can no longer stain the crystals with DNA. So we think they actively bind, the DNA actively binds to this repetitive carbohydrate recognition domain array on the outside of the crystals. And in this way, the whole thing gets tangled. And this really matters for the quality of the mucus. So we can measure the quality of the mucus using rheometry, which is a kind of a plate that deforms the mucus and then lets it relax. So you measure the viscosity and the elasticity of the mucus gel. So if you take a mucus gel and you add neutrophils to it, there's not much happening to the elasticity. However, if you add crystals to the neutrophils, you see that there's an enormous increase in the elasticity of the mucus. So in other words, it starts behaving like a glue. When you take a dry glue and you pull on it, it's going to return to its original position. This is exactly what is happening. But if you neutralize the DNA, you degrade the DNA with pulmozyme, it's a drug that you can use via inhalation. The pulmozyme normalizes the change in elasticity. It's actually showing that the DNA that's triggered by the sarcolytin crystals from the neutrophils alters the stickiness of the mucus. So this idea of barbed wire accumulating in the mucus is another factor how crystals can do harm. So the crystals are in the mucus plugs, and when there's a neutrophilic exacerbation, which you often see with viruses, remember viruses are the most common cause of asthma exacerbation. When those neutrophils come in, they will see the crystals and they will go completely crazy. Just like in gout, when neutrophils see uric acid crystals, you get this massive swelling on your toes. The same thing will happen in your airways. You will have a massive swelling of the mucus plug. And we believe there's a case like this from our colleague Ueki in Japan. So this was a child coming in with influenza A virus infection, no prior history of asthma, comes into the ER with an upper lobe that's completely out of air. 30 minutes later, the entire left lung is out of air. They do a bronchoscopy, a mucus plug comes out, and it's full of sarcolytin crystals that can be stained with galectin-10. So what happened in this particular case is the kid had a pre-existing plug, had asthma, but didn't know it. But then the neutrophils came in response to the influenza A virus, and this caused an immediate swelling and growing of the plug so that there was a complete obliteration of the lung in a couple of minutes happening. And we believe this is also happening in patients with asthma exacerbations. So the last couple of slides I want to show is we want to take this to the clinic. I've shown you some molecular biology, some mouse work, some human work, but the proof of the pudding is in the eating. You know, we have to work together with biotech to come up with drugs to target this. So we teamed up with a biotech company, Argenix, here in the neighborhood here in the same city, and very early on we were able to convince them to make antibodies to sarcolytin protein. So we immunized llamas, and then generated from the heavy chains and the light chains, generated a phage library so that we can screen for binders to galectin-10. And then once we had binders, we reformat them to conventional antibodies. And when you take these llama antibodies and you put them on preformed crystals, you can completely shrink those crystals. I will run the video again. The crystals shrink along the long axis. And the same thing when you take this highly elastic mucus out of the nose of a patient. So you see it's full of crystals here, sarcolytin crystals here, also these big rods here. So if you put antibody to galectin-10 on these crystals, you see the small crystals shrink in the time frame of a couple of hours. And also this large material, which pathologists would call junk or amorphous material, is no such thing as junk. It's a crystal of sarcolytin protein of galectin-10 that can completely be solubilized when you put antibody on that crystal. So how does it work? The antibodies actually bind to the tyrosine 69 that's available on the tip end of the crystal. So this is the antibody here, the heavy chain of the antibody sticking out a tyrosine to the tyrosine 69 of galectin-10. So it's a pure molecular mimicry so that you can compete with galectin-10. So it's just like Lego. If you're gonna break down your Lego, you're not gonna start in the middle, you're gonna start at the tip ends where it's still loose, and you're gonna chew up your blocks, you know, from your Lego piece, and like this gonna make it smaller again. The same thing happens with these antibodies. They bind to the key crystal packing interface and go in competition, and they solubilize the crystal. And these antibodies also work in vivo. So this is a humanized asthma model where you see the goblet cell metaplasia, the MUC5AC production induced by crystals. And if you treat these animals with an antibody that solubilizes the crystals, we can get rid of the goblet cell metaplasia, the MUC5AC, and the human IgE production that's typical of this model. So the last 2-3 slides will be on other protein crystals doing this. So remember, the mouse doesn't have sarcolytin crystals. However, mice also have crystal, and these are very old slides, again, from 1964. These are airways of mice where you see things that completely look like sarcolytin crystals. So you have these needles sticking into the epithelium, in the mucus, impinging on the epithelium, and being trapped in mucus. And some of these crystals are needle-shaped and some are plate-shaped. So what are they made of? So we've developed immunostainings also. These crystals are made up not of galectin-10, they're made up of YM1 or YM2. So these are chitinase-like proteins. They're also known as CHIL3 and CHIL4. And CHIL3 is mainly made by macrophages, alternatively activated macrophages and neutrophils, whereas CHIL4 or YM2 is made by the lung epithelium. And there's no doubt that in mice with asthma, you can find these crystals very, very easily. So we can also recombinantly make these crystals. So we have pure biosimilar YM1 and YM2 crystals. And just like with Charcot-Leyden crystals, Ines Hendricks in our lab has been able to show that we can trigger monocytosis, eosinophilia, IL-6 production, TNF-alpha production, particularly with the crystalline state, but not with the soluble YM1. So the crystalline state of these proteins determines whether they're pro-inflammatory. And just like with Charcot-Leyden crystals, these crystals can be taken up by dendritic cells. This is here the new way of gating. It's Kavita Rawat who published this. So it's C11C, C11B, broad gate. Then you gate your monocytes and your conventional DCs based on CD88, CD26, CDC1, CDC2s. And you see immediately that the crystals trigger the recruitment of monocytes and CDCs to the airways. And if you then co-administer fluorescent ovalbumin, it's mainly the CDCs that have taken up antigen that also mature and become CD86 positive. So dendritic cells mature, and when you mix them with ovalbumin, just like with the Charcot-Leyden crystals, these YM1 crystals boost the proliferation of OVA-specific T cells compared with soluble OVA or soluble YM1 plus OVA. And this leads to Th2 development. So here we crossed the OT2 T cells that are increasingly found with a CD4, an IL-4, KN2 reporter mouse. And we see that those Th2 cells develop, those T cells develop into Th2 cells only with the crystalline OVA, but not with the soluble OVA. And they return to the lungs as effector cells. And when we re-challenge those mice with OVA aerosol, we get eosinophilia. So this is YM1. There's also YM2. So YM2 is mainly made by lung epithelial cells. So this is an organoid that Ursula Smolle made. It's a mouse organoid from lung basal epithelial cells that develops into beautiful airways in vitro. There's no other cells but epithelial cells in there. If we throw IL-13 on there, even in the organoids, we see crystal formation. So this is a crystal that was fished out. We got crystal structure out of that organoid. Again, Kenneth Verstraete and Savas Savides. And these are YM2 crystals. And we believe that YM2 also has a form in solution. So we've made knockouts of YM2, soluble YM2. And we actually see that YM2 as a soluble protein has the potential to boost type 2 immunity. So if we do innate immunity to IL-33 by giving mice 3 days of IL-33 to the airways, we can see IL-5 production and ILC2s. But when we mix this with YM2, we get a severe enhancement of the response to innate IL-33. So we get more bowel fluid IL-5, more bowel fluid ILC2, more amphiregulin production. And we believe this is due to activation of alveolar macrophages. So this is a single cell analysis of both the stroma as well as the immune cells. And suffice it to say that when we give the combination of IL-33 plus YM2, this is here just focusing on CCL17, a known chemokine for ILC2s, we see that in the PBS situation, alveolar macrophages make very little CCL17, but Ursula found that in the combination of IL-33 plus YM2, the AMs start making CCL17. This is also on sorted AMs. The mRNA is there. The protein is there. And when we look at the recruitment of ILC2s, this is blocked by blocking CCL17 chemokine. So YM2 has also, and as a soluble molecule, a potential to promote type 2 immunity. So in conclusion, I've shown you 2 examples of proteins that crystallize. They both do it in type 2 immunity. And here comes the funky thing. In humans, this is done with an eosinophil-derived protein, galectin-10, leading to Charcot-Leyden protein formation. Mice do not have galectin-10. Mice do it with another pathway. They use alternatively activated macrophages to produce YM1 crystals, and humans don't have YM1. So it's like evolution has found a solution to the same problem, but with different proteins coming from different cell types, but highly typical in type 2 immunity you form these crystalline structures. And we think they're very potent in triggering mucus, triggering dendritic cells, triggering ILC2s, causing netosis. And this is all important, we believe, for anti-helminth defenses. But as a corollary, you see this as a bad sign in asthma. We're also thinking more and more that those crystals could act as a depot, slowly releasing crystals that might have functions also as soluble proteins. And it could even be pro-inflammatory, it could be pro-repair. So this is the group of people working on this, and I particularly want to point out Hamida Hamad, who's been my colleague for the last 20 years, set up many of the house dust mite models. And this is the crystal team. All these people are working on various aspects: Helena Eigerter, Ines Hendricks, Angie Ahmed, Jasmijn Burgelman, and Dora Parasai. So thank you very much for attention. I'll be happy to answer questions via Twitter or X. Thanks.
Yeah, thank you so much, Bart. This was an outrageously fantastic seminar, as usual, exactly what I expected. So thanks so much for delivering this. And I'm sure there will be a lot of questions. I just realized it's no longer called Twitter. We still call it Twitter, but I guess, yes, it's on X now. There will be plenty of questions for you. It'd be good if you could keep an eye on your X handle and answer those questions. So please don't be shy. Ask Bart as many questions as you like, and keep him busy for a while after this fantastic seminar. With this, I would like to thank everyone for, again, coming to Global IMMUNOTALKS, listening to us, listening to Bart, your support, and I wish everyone a wonderful morning, afternoon, evening, night. Bye-bye, everyone.